Alright, we are done with catabolism, the second metabolic task. And before we continue talking about metabolic tasks, let's once again talk about how cells make ATPs, or adenosine triphosphate molecule. We already know one way that cells use to make ATPs, which is a substrate level phosphorylation reaction. Remember, glycolysis and the TCA cycle actually use a substrate level phosphorylation reaction to make ATPs. But there's another way for cells to make ATPs, and please remember, most ATPs in a cell are made by this way. The name of this way is chemiosmosis. Chemiosmosis. On this slide, you see plasma membrane, bacterial plasma membrane. At the bottom of this picture, this is the area of cytoplasm of this cell, and on the top of this picture you see the area which is outside of the cell. There is a compound which is embedded in between two layers of plasma membrane. Remember, plasma membrane is bilayer. It's made of two layers. So electron transport chain compound, or ETC compound, is embedded in between two layers of plasma membrane. Also, let me remind you that all catabolic pathways produce a lot of NADPH molecules. Those NADPH molecules actually are used for reaction phosphorylation. They help to convert ADP molecule, which is low energy molecule, into ATP, which is a high energy molecule. Chemiosmosis begins when two hydrogen atoms from an NADPH molecule are transferred to electron transport chain. Let me remind you, hydrogen atom is composed of two parts, proton and electron. When hydrogen atoms are transferred to electron transport chain, only electrons will be accepted by this chain, but protons will be pumped out outside of the cell. When this reaction repeats several times, continues several times, or occurs several times, a lot of protons will be pumped out outside of the cell. As a result, they create what we call proton gradient. Means we have a lot of protons outside of the cell and not too many in cytoplasm. Next step is protons from outside of the cell will try to get back into cytoplasm. The only way for them to go across the plasma membrane is to go through what we call proton channel. This is another compound which is embedded into plasma membrane. On this picture you see proton channel in green color. When protons go through proton channel, because they have positive charge on them, they create a lot of energy. All that energy will be picked up by ATPase, which is enzyme of proton channel, and then it will be used for reaction phosphorylation. So this way two ADP molecules will be phosphorylated with two PO4 groups, phosphate groups, and as a result that ADP molecules will be converted into two ATP molecules, which are high energy molecules. Let's repeat it once again. So chemiosmosis begins when two hydrogen atoms, remember each hydrogen atom is composed of one proton and one electron. So two hydrogen atoms are transferred to electron transport chain. But electron transport chain accepts only electrons, but protons will be pumped out of the cell. Let me remind you, all of these are chains of biochemical reactions. When a lot of protons get accumulated outside of the cell, they form what we call proton gradient. Next, protons go through the proton channel back into cytoplasm, and as they go through the proton channel, which is also embedded into plasma membrane, they create a lot of energy. That energy will be picked up by ATPase enzyme of proton channel and will be used for reaction of phosphorylation of ADP molecule. But what is going to happen to electrons that are picked up by electron transport chain? Electrons will be moved to the end of electron transport chain where they will be picked up by what we call terminal electron acceptor. We are talking about aerobic metabolism right now, or aerobic respiration. For aerobic cells, the terminal electron acceptor is always oxygen. So oxygen picks up electrons at the end of the chain and then will be reduced into water. So the terminal electron acceptor for aerobic microorganisms is oxygen. How about anaerobes? Anaerobes do not use oxygen for metabolism, right? That's right. For anaerobes, terminal electron acceptors will be sulfates, nitrates, and fumarates. The next slide, this slide shows you where all catabolic pathways, including chemiosmosis, ETC, electron transport chain, occur in eukaryotes and prokaryotes. Please, you have to memorize this chart for your coming exam. Let's start with glycolysis. The first catabolic pathway. You see in eukaryotes, glycolysis occurs in cytoplasm. Same is in prokaryotes. Intermediate step. We already know in eukaryotes it will occur in mitochondrial matrix, and in prokaryotes cytoplasm again. Why? Because prokaryotes have no mitochondria. TCA cycle or Krebs cycle. In eukaryotes, once again, it occurs in mitochondrial matrix, in prokaryotes, in cytoplasm. And finally, chemiosmosis or ETC. In eukaryotes, it will occur in mitochondrial inner membrane, and in prokaryotes, remember, ETC is embedded between two layers of plasma membrane. On this slide, you see a picture of a mitochondrion, and as you see, it's made of two membranes. This is what we call outer membrane, and this is a structure that we call inner membrane, and it forms what we call crista. So please pay attention. ETC, electron transport chain in eukaryotes, is embedded into inner membrane somewhere over here. This slide shows you how many ATPs produced by prokaryotes and eukaryotes. As you see, prokaryotes make 38 ATPs versus 36 ATPs produced by eukaryotes. The question is why prokaryotes make more ATPs than eukaryotes. The reason why, because prokaryotes have higher metabolism, faster metabolism, and we're going to talk about it next lecture. In this chart, they show you how many ATPs are produced by substrate level phosphorylation. You remember glycolysis makes ATPs, Krebs cycle makes ATPs 4 together, but the rest of ATPs are made by chemiosmosis. Remember I told you cells produced most of ATPs by chemiosmosis.